So today we are going to talk about two major phenomena of respiration. Number one is the chloride shift and number two is the reverse chloride shift. So look at them. So as you all know that carbon dioxide is transported in our body via three major ways. Number one, in the form of bicarbonate ion. Number two, in the form of carbaminohemoglobin. Make sure you don't write carboxyhemoglobin. It's carbaminohemoglobin. Carbaminohemoglobin. And lastly, in the form of carbonic acid. This constitutes almost 70% of the transport. This one 25%. And this one just 5%. Now, there are two major processes, chloride shift and reverse chloride shift, are indulged in the transport of all of these ions via carbon dioxide. So let's see. Number one is the chloride shift. Chloride shift, also known as the hamburger effect. Also known as the hamburger effect. And second one is known as the reverse chloride shift. Reverse chloride shift, also known as the Halden effect. Now we are going to study about both of these in detail. So now we are going to start about chloride shift mechanism. Okay, so this one is our epithelial tissue of our blood vessels. Let's suppose that this one is our RBC. This one is our metabolizing tissue. Now what we all know that carbon dioxide is released by a metabolizing tissue and in turn oxygen is given by RBC to the metabolizing tissue. So what we have in this RBC? First of all, we have carboxyhemoglobin, HbO2. We are having water, H2O. And here we are having sodium chloride ions. Now, as the metabolizing tissue is giving out lots and lots of carbon dioxide to it, it will ultimately penetrate the epithelial tissue and move into the red blood cell. And there it will combine with the oxygen. This carbon dioxide will combine with the water and will form H2CO3. This one is carbonic acid, right? This one is carbonic acid. Now this carbonic acid in the presence of another enzyme, carbonic anhydrase. Carbonic anhydrase will dissociate it into H plus and HCO3 minus ions. Now as you can see, there is an increasing concentration of hydrogen here. That means there is a constant reduction out here. So what will happen? This carboxyhemoglobin will be dissociated and reduced hemoglobin will be formed. So see what's happening. H.Hb is being formed and this carboxyhemoglobin will release oxygen and this oxygen will in turn be given to the tissues. Now the oxygen will in turn be given to the tissues. Now our motto is accomplished. We have taken the carbon dioxide from the tissues and in turn gave oxygen to it. Now where is chlorine here? Now see what is the fate of this bicarbonate ion. Now one thing you must know that this membrane is specifically permeable to anions and not cations. Specifically permeable to anions and not cations. Hence here there are lots of Na plus and Cl minus ions. When the carbonate ion will come out of this red blood cell, another phenomenon is going to occur, which is known as the electrostatic imbalance. As the bicarbonate ion will come here, there will be electrostatic imbalance in this position, Hence there will be a rapid influx of chloride ion into the cell. There will be a rapid influx of chloride ion into the cell. See, there is the chlorine is constantly moving into the red blood cell. So there is a rapid influx of chloride ion, hence this phenomenon is known as the chloride shift. And what will happen to this bicarbonate ion? It will eventually combine with the sodium and form sodium bicarbonate. Now make sure that this phenomenon, where it is occurring, it's occurring near the metabolizing tissues, that means body tissues. Here three phenomena are occurring. Carbon dioxide is being released from the tissue into the red blood cell. Oxygen is being given to the tissue, and chloride. There is a shift of chloride ion from the plasma inside the red blood cell. Hence this total phenomenon is known as the chloride shift. Hence this total phenomenon is known as the chloride shift. Now take a note of this, then we will go for the reverse chloride shift. There the reverse process will occur where the chlorine will be given out by the cell. So here we are starting our next phenomenon, which is known as the reverse chloride shift. Now if you guys remember that previously in the red blood cell what we had? We had carbaminohemoglobin, which was formed due to the rapid influx of carbon dioxide from the metabolizing tissue. Also we had reduced hemoglobin. Now this same RBC is now traveling to closer to the alveolar tissues. Now we all know that as we are inhaling the oxygen, the alveolar tissue is filled with oxygen. So there will be a rapid influx of oxygen via an epithelial tissue into the red blood cell. As there is rapid influx of oxygen, then this reduced hemoglobin will combine with this and form oxyhemoglobinic acid. Make sure that this hydrogen is still attached to it, and it's oxyhemoglobinic acid. Now this oxyhemoglobinic acid will try to reduce, this one is a strong acid, it will try to dissociate this carbaminohemoglobin, it's not carboxyhemoglobin, it's carbaminohemoglobin, into Hb plus CO2. Now this CO2 will be in turn given back to the alveoli, from where it will be exhaled out by us. Now what is happening out here? As the concentration of carbaminohemoglobin is decreasing, this oxyhemoglobinic acid will also lose its stability, and it will form H plus and carboxy hemoglobin. Now seeing this H plus inside, this HCO3 minus outside the cell will go inside. As you know that there are also chloride ion inside. From the previous process there are chloride ions due to the chloride shift. Now as this HCO3 minus will enter this cell, immediately the chloride ion will come out of the cell, and this is known as a reverse chloride shift. Now you can see that here the chloride ion is being transferred from the cell to the plasma. It's just the reverse of the previous one, where it was being transferred from the plasma into the cell. This one here, chloride ion is being transferred from the cell to the plasma, and this HCO3 will combine with hydrogen and form H2CO3. This one is carbonic acid. Now this carbonic acid, in the presence of that same enzyme, carbonic anhydrase, will break out into, let me dump this carbonic acid will break into, in the presence of carbonic anhydrase, it's a very important enzyme, H plus, H2O and CO2. This CO2 will again be transferred to the alveolar tissue where it will be exhaled. Now you can see that it is just the reverse of the previous one, where the chloride ion is shifted from the cell to the plasma. This one is known as the reverse chloride shift. Now what are the main points of this phenomenon? The main points of this phenomenon is that there is a rapid influx of oxygen from the alveoli into the cell. there is a rapid efflux of carbon dioxide from the cell to the alveoli, from where we will exhale it out, and then there is a shift of chloride atom from the RBC to the plasma, and it will in turn form NaCl outside. So this is the major phenomenon of both reverse chloride shift and chloride shift. Now we will summarize the points as we make out the difference. Just take a note. Now we are going to make out the difference between the two processes. Number one is the chloride shift. Also known as the hamburger's phenomenon, and second one is the reverse chloride shift. Also known as the Halden method. Now first of all, who discovered both these phenomena? This phenomenon was discovered by J.H. Hamburger. Discovered by J.H. Hamburger, and from there it got its name Hamburger's. This was discovered by John Scott Halden. Discovered by John Scott Halden. So from where? Now in this method we needed carbon dioxide. There was a rapid influx of carbon dioxide. So you think from where will this method take place? Yes, this method will take place near the metabolizing tissue. Takes place near metabolizing tissue. Where does this method take place? Here we needed oxygen. From where did we get that? We have got that from the alveoli. Takes place near the alveoli of the lungs. Here there was a reductive environment, that is carbon dioxide was taken in my cell, rapid influx of carbon dioxide from the metabolizing tissue. If you guys remember, you put influx of CO2 from the cell, from the metabolizing cell, from the cell to the RBCs, and there is a rapid influx of oxygen. Here it's carbon dioxide, and here rapid influx of oxygen from alveoli. You take these points, and lastly we write the most important thing that's going to happen, that is the chloride shift. This is the most important point. As the name suggests, there is a shift of chloride ions. There is a shift of chloride ions from the blood plasma into the RBC. Shift of chloride ions from the blood plasma into the RBCs. Into the RBCs. Fourth point here, the reverse will, there will be the shift of, there is a shift of chloride ions from where to where? Just the reverse of it, from the RBCs to the blood plasma. Shift of chloride ions chloride ions from the RBC to the blood plasma. So that's all for both the processes. I hope you people have understood. So thank you for watching. All the very best.